Hey guys, thanks for joining me again for another episode of my YouTube channel, Long Live the Brains. Today is April the 19th and we're still in Parkinson's Disease Awareness Month. So I'm going to continue talking about all things Parkinson's disease and today focus on the genetics of Parkinson's disease. Now in video 42, I addressed the question of, is it genetic? And briefly, whenever I get asked this question, which is very common, essentially what people are trying to understand is, if my mother or father have disease X, be it Alzheimer's disease or Parkinson's disease, what are the chances that I'm going to develop the disease, or what are the chances that my children are going to develop the disease? And what we may have thought a few decades ago was going to be a simple question, yes, this is genetic, or no, this isn't genetic, has actually become much more complex. And I dug into some of those complexities in video 43 where I discussed the genetics of Alzheimer's disease. But today I'll focus on the genetics of Parkinson's disease. Before I do that, I want to touch a little bit on the basic science about proteins and DNA and where everything fits. And I've discussed some of this in previous lectures. Video 39 discussed proteins and how they're molecular machines. They're critical for performing all types of cellular functions in our body. They're the machines that do all the heavy lifting. So without them, we can't live, can't breathe, can't do anything. But like all machines, even in real life, to build a machine, you've got to have instructions. And those instructions are found in our DNA. Now that DNA is broken down into genes, and it's those genes that are then transcribed into a similar compound called RNA, and then that RNA is what's translated into the protein or built into the protein. All right. So if you have a problem with the instructions, that being the DNA, then you're not going to get the appropriate machine. There might be a defect in the machine. And if there's a defect in the machine, then it's not able to carry out its designated function. And if it can't carry out its designated function, then we develop symptoms or disease. Okay, so that's why there's all this interest in genetics, because by understanding where in the instructions things are going wrong, we understand what protein is dysfunctional and where that protein fits into a biochemical cascade of cellular processes. And so we can understand how the disease is working, and we find potential therapeutic options. So if you can find a protein that's abnormal, then you can target it potentially with a drug. And so all of these steps are complex and require a lot of attention and money on the part of research scientists and grants. But they're absolutely critical to our understanding of the disease and the development of better treatments and cures. Okay, so we know that the proteins are potentially druggable targets. It's something that we're aiming to find so that we can create new treatments. But we have to identify, to be able to identify these proteins, we need to identify where the gene is, that if it's mutated, the instructions are bad, causing the machine to be bad. So we need to identify the gene. And that's a very complex problem. So think about how you would identify or how you would locate me, okay? So I would give you my address and you should be able to find me. Now, what if I just gave you the city that I'm in? I'm in Jacksonville. Well, that's a start. That helps to knock out other states and other cities. You know that I'm in Florida. You know that I'm in Jacksonville. So think about chromosomes. DNA is within chromosomes, and we all have 23 chromosomes. We actually have 2 pairs of those chromosomes. So companies like 23andMe, that's where the 23 comes from. It's referring to our 23 chromosomes. So finding where the problem is in terms of which chromosome it's on is helpful, but it's by no means the whole story. So that's like me telling you I'm in Jacksonville, good luck, go find me. That's not easy to do. So what if I gave you my street that I'm on? Well, that would help narrow things down quite a lot. So in the field of genetics, we narrow things down to the chromosome, to which chromosome we're dealing with, and then within the chromosome, a specific locus, or that's sort of a region. It's kind of like your street, what street you're on. Now, there are lots of people that may live on your street, lots of houses on your street, and those are kind of like the genes that are within the locus. So there are many genes within a locus. So initially, when we are trying to nail down and find a gene, we can find a locus, and then we have to start testing to figure out which gene it is that we're picking up a signal from. So which house, okay? So if I told you my full address, you'd have the city, so the chromosome. You'd have the street, so the locus. And you'd have the number on the street, and that would tell you what gene we're talking about, okay? So in the context of Parkinson's disease, we initially identified loci, or that's the plural for locus, and those are designated by Park and a number. So Park 1, Park 2, Park 3, Park 4, so on and so forth. Okay, so now let's dig into Parkinson's disease. If you were to ask someone 30 years ago if Parkinson's disease was genetic, they would probably tell you no. And that was based largely on studies that were done in twins, and the evidence just wasn't there to support a genetic cause for Parkinson's disease. However, now in 2019, we know that about 15% of patients with Parkinson's disease have a family history of Parkinson's disease. Okay, so in 1997, actually I should back up just a little bit. In 1996, we found Park 1, or the locus, the first locus associated with Parkinson's disease. Now, I should say that as we found more and more loci, most of these are associated with young onset Parkinson's disease. So when we talk about something that's pretty clearly a heritable Parkinson's disease, it's usually going to present younger. In the case of Park 1, we're talking about maybe even as early as the 4th decade of life. Okay, so 1996, we found Park 1. And then just a year later, we were able to narrow it down to the gene, and that's the SNCA gene. Now, why is that important? Well, we learned that SNCA on the DNA, remember, it's the instruction, eventually it's translated to create synuclein. Now, synuclein is an abnormal protein that's deposited in the brains of patients with Parkinson's disease, dementia with Lewy bodies, multiple system atrophy, and that's why we refer to these diseases collectively as alpha-synucleinopathies. So this shows sort of the process behind finding the gene, the instruction, all the way to the protein that we know is present in abnormally high amounts in patients with Parkinson's disease. Fast forward about a year or so later, we identified the second locus and then gene, that being the Parkin gene. And then a few years later, we got Park 3 and Park 4 and Park 5 and Park 6. Park 6 is Pink1. And Park 7 is DJ1, that's another gene. And Park 8 is LRRK2. Now, I mentioned those 5 genes, SNCA, Parkin, Pink1, DJ1, and LRRK2, because these are kind of the heavy hitters when it comes to Parkinson's disease genetics. SNCA and LRRK2 are both inherited in an autosomal dominant pattern, meaning if you have that mutation, the disease from the mutation, then your child has about a 50% chance of having that mutation and subsequently the disease. Now, the other 3 genes that I mentioned, Parkin, Pink1, and DJ1, those are typically inherited in an autosomal recessive fashion, meaning that to actually have the disease, you've got to have a copy of that mutation on both alleles of that chromosome. So what that essentially translates into is about a 25% chance for offspring to develop that disease. And again, I'll refer you back to video 42 entitled Is It Genetic?, which digs into autosomal dominant and autosomal recessive inheritance pathways for more details there. You know, the big buzz about genetics, it's not simply, you know, just for academic exercises. We want to find the DNA, find the gene, so that we can figure out why there's an abnormal protein every step along the way. Not even just the protein, but potentially even the RNA and even potentially the DNA provide potential therapeutic targets for drug development, because the name of the game, again, is developing better treatments and ultimately cures.